Hi, everybody. I'm going to run through pages 27 through 31 of your interactive notebook. We had you start doing a case study, and unfortunately, it didn't work as well as we wanted to, but hopefully, as you were kind of watching Miss Ramsey go through it, you got a sense of what was going on with this dog, Claire, right? So you were asked to imagine yourself as a vet tech, and this dog, Claire, comes in, a Great Dane, and has some symptomology, right? Has some symptoms rather um, that are severe, right? Extreme weight loss, lethargy, eating but not maintaining weight, and so this leads you to believe there might be some issues with Claire's digestion and metabolism, right? And so you can see the definitions of those two things there, and you probably had some sense of what those were. And a large part of digestion, right? Certainly it starts with mechanical aspects, right? Chewing, crushing, but right away, right? Right in your mouth, right? You have amylase, an enzyme that starts acting to help speed up chemical reactions that allow you to digest foods that you're eating. And this is the same in any organism like Claire, right? And so a couple of the enzymes and where they're located, remember there's thousands of different enzymes, but these are some of the important ones and where they're located: pepsin, protease, carbohydrates, lipase, most of them, of course, ACE at the end. And here's what they do, right? Shouldn't surprise probably anybody that carbohydrates breaks down carbohydrates, right? Lipase breaks down lipids. Okay. So your job was essentially to determine what was going on with Claire by running some tests and seeing if you could explain sort of what was happening. And so some of the potential issues that could be affecting Claire's digestion, a mutation, some issues with pH, low enzyme production. And think about what all of these things do, right? All of these things have the potential to throw off your homeostasis, right? That ability to sort of balance and do what a cell needs to do. And so enzymes can play a big role in that. You run some process of elimination tests. You can see here. So basically, you rule out the mutation, you rule out the abnormal pH because those tests came back normal, but it looks like the concentration of enzymes are lower than they should be. So why is this, right? Why is Claire not making the enzymes she needs to? Well, she has this insufficiency, right? This EPI, and the cells of the pancreas become damaged and they stop secreting a high enough concentration of digestive enzymes to be effective, right? So just like if you're lactose intolerant, right, you can take lactase or you can buy milk that's already had the lactose broken down. Sort of the treatment here with Claire is to give her enzyme supplements, right, that already contain these ones, protease, lipase, and carbohydrates, so that when she consumes food, right, she'll have the ability to break it down. Her metabolism will have the capacity to make her healthy again. So this is a real issue, right, that could happen. In fact, you're starting to hear about EPI in humans now, too. So just one illustration of how enzymes can be really important. Okay, our last macromolecule. Remember, to this point, we've talked about carbohydrates, we've talked about lipids, we've talked about proteins, and of course, under a subset of proteins are the enzymes. The last one we need to talk about here are the nucleic acids, right? Obviously, a very important macromolecule for living things. Again, we see carbon, we see hydrogen, we see oxygen, but we're also adding some nitrogen and some phosphorus at this point, right, into the structure. The function of this stuff, what does it do for us? It stores and transmits genetic information. amongst other things, right? There's actually a few other things that it can do, but we'll get into that actually when we get into photosynthesis and cellular respiration. So what are the monomers? Sorry about that. What are the monomers? It's essentially made of a 5-carbon sugar, a phosphate group, and a nitrogen base, and you can kind of see that in the picture here. And those are in turn strung together to make increasingly larger sections of either DNA or RNA. In humans, the human genome, you see these little things that are across from each other, this green and this red and this blue and this yellow, those are called base pairs. In the human genome, we have 3.2 billion of those in each of our cells, actually. And so this stuff's pretty important, and there's a lot of it in our body. So we had you do a quick little virtual lab type thing where you were able to piece some together. You were to build a complementary piece of DNA. That was the Gizmo lab. And I think you posted that to Padlet, hopefully. I'll look here. I saw some popping up here just recently, tonight. Okay, finally, we had you look at a virtual DNA extraction, and honestly, anything that's living, or the vast majority of things that are living, you should be able to extract DNA from, right? So if it has a cell and it has a nucleus, it's got DNA inside of it. Viruses can sometimes be RNA based, but even those are largely DNA based. So anything that's cellularly based is going to have DNA in it, so you should be able to extract the DNA from it. Why would you want to do that? There are lots of reasons for looking at the DNA to see if there's any issues, genetic diseases potentially underlying, for identifying bodies, right? We have sort of a fingerprint, forensic evidence, that type of thing. Again, the DNA is in the cells. You can get it out of your own cells. You can get it out of bananas. You can get it out of strawberries. Those tend to be fairly easy because they have a lot of DNA. They're polyploidy, which means they have lots and lots of copies of their DNA. But the process is really the same, right? You essentially need materials that can break down what a cell is made of. And we know now what a cell is made of, right? It has an outer layer of lipids, right? A phospholipid bilayer that surrounds its membrane. So you have a detergent that helps break that down. You want to add an enzyme to help cut away some of the proteins from the DNA to expose the DNA. So your goal is really to get DNA to sort of clump together. You're kind of trying to create the right chemical environment to get it to clump together. Hopefully you've had a chance to do this. Maybe we can demo it in class at the bare minimum. But at the end of it, you get some DNA that looks kind of, I don't know, for lack of a better way of saying it, I guess sort of like snot, a white sort of material like that. Okay, so tomorrow we will review for the Unit 2 summative assessment, which will be on Wednesday. We'll see you.